Good day everyone! I am Mabibilan Nasabal, report for this topic, Gametogenesis. So for you, what is Gametogenesis? Any idea? Gametogenesis is Gamos means marriage, genesis means origin, is the produce of gamete formation in the sexually reproducing animals. The sexually reproducing animals contains two types of cells in their body, somatic cells and germinal cells. Both types of cells diploid number of chromosomes, but each type has its different destiny. The somatic cells form various organs of the body and provide a phase for the maturation, development, and formation of the germinal cells. The somatic cells always multiply by mitotic division. The germinal cells form the gonads, testes, ovaries in the body. These cells produce gamete cells by successive mitotic and meiotic division. The male gamete is known as spermatozoon or sperm, and the female gamete is known as vacuum or egg. The process of sperm production is known as the spermatogenesis. Sperma means sperm or seed, genesis means origin, and the process of production of vacuum is known as oogenesis. Oogenesis, oo means egg, genesis means origin. Both the process can be studied in a detail under separate headings. Gametogenesis has two types, in which spermatogenesis and oogenesis. Spermatogenesis is a formation of sperm in testes, and oogenesis is a formation of egg in an ovary. Comes into your mind, what is spermatogenesis? So let us define. Spermatogenesis is the process in which spermatozoa are produced through mitosis and meiosis from male primordial germ cells. As spermatogenesis occurs in the male gonads or testes. The testes of the vertebrates are composed of many seminiferous tubules, which are lined by the cells of germinal epithelium. The cells of the germinal epithelium forms sperm by process of spermatogenesis. But in a certain animals, mammals, and mollusca, etc., there are somatic cells lying in between germinal cells. The somatic cells are known as Sertoli cells. The Sertoli cells anchor the differentiating cells and provide nourishment to the developing sperms. The insect do not possess Sertoli cells. When diploid spermatogonia form haploid spermatozoa, the process is called spermatogenesis. Spermatogenesis is divided two major whence the first one begins in the formation of spermatids and spermiogenesis. Formation of spermatids, the male germinal cells which produce the sperms are known as primary germinal cells and primordial cells. The primordial cells pass through following three phases for the formation of spermatids. 1. Multiplication phase, 2. Growth phase, and 3. Maturation phase. Now, let us study the multiplication phase. In this step, the mitotic division of primordial germ cells, spermatogonium A, takes place and gives rise to spermatogonium B cells. These spermatogonia cells are generated from the germinal epithelium of the seminiferous tubules. The spermatogonia grow in the size and differentiate into primary spermatocytes. 2. The growth phase. In the growth phase, the spermatogonial cells accumulate a large amount of nutrition and chromatin materials. Now, each spermatogonial cells is known as primary spermatocytes. Maturation phase. The primary spermatocytes are ready for first meiotic or maturation division. The homologous chromosomes start firing synapses. Each homologous chromosome splits longitudinally and by the chiasma formation, the exchange of genetic material or crossing over takes place between the chromatids of the homologous chromosomes. The DNA amount in duplicate in the beginning of the division. The first division of primary spermatocytes is the reduction division, meiosis. This results in two daughter cells, one with 22 plus X chromosomes and another with 22 plus Y chromosomes. These are called secondary spermatocytes. Then the second meiotic division, meiosis II, takes place, which form four daughter cells known as spermatids. The spermatids gets matured into the sperm cells, spermatozoa. This process is known as spermiogenesis. The release of spermatozoa cells into seminiferous tubule is called spermiation. 2 is spermiogenesis. The metamorphosis or differentiation of the spermatids into the sperms is known as spermiogenesis because the sperm of spermatozoon is a very active and mobile cell, so to provide great amount of mobility to the sperms. The superfluous material of the developing sperm is discarded. For the reduction of the weight of the sperms, following changes occurs in spermatids. Changes in the nucleus. 2. Acrosome formation. 3. The centrals. The anterior one is known as proximal central and posterior one is known as distal central. So, spermatogenesis is a process. Changes in the nucleus. The nucleus loses water from the nuclear substrate and assumes different shape in the different animals. The sperm nucleus in a man and bull becomes ovoid and laterally flattened. In rodents and amphibians, the sperm's nucleus becomes scimitar shaped with pointed tip. In birds and mollusks, a nucleus becomes spirally twisted like a corkscrew. The bivalve mollusks have the round sperm monoculus, and shape of nucleus also determine the shape of the sperm head, which becomes fully adapted for the active propulsion through the water. The RNA contents of nucleus and the nucleus are greatly reduced. The DNA becomes more concentrated and the chromatin materials becomes closely packed into small volume. So this is a peak of spermatogenesis and oogenesis processes. 2 is acrosome formation. The acrosome occurs at the anterior side of the sperm's nucleus and contains protease enzymes which help it easy penetration inside the egg. The acrosome is formed by the Golgi apparatus. The Golgi apparatus is concentrated near the anterior end of the sperm nucleus to form an acromosomes. 1 or 2 volculus of the apparatus become large and occupy the place between the tubules of Golgi apparatus. Soon after, the dense granule known as the proacrosomal granule develops inside the vacuole. The rest of the Golgi apparatus becomes reduced and discarded from the sperm as Golgi rest. In the sperm of certain animals, an acrosomal cone or axial body also develops in between the acromosome and the nucleus. So let's proceed the centrals. The two centrals of the spermatids become arranged one after the other behind the nucleus. The anterior one is known as proximal centrals and the posterior one is known as distal central. The distal central changes into the basal bodies and gives rise to the axial filament of the sperm. So, makikita nyo dyan ang axial filament in this picture. So, the axial filament or the flagellum is composed of 4 central longitudinally fibres and 9 peripheral fibres. The distal central and the basal part of the axial filament occurs in the middle piece of the spermatozoa. The mitochondria of the spermatids fuse together and twist spirally around the axial filament. Thus, most of the cytoplasmic portion of the spermatids except the nucleus, a chromosome, centrals, mitochondria, and axial filament is discarded during the spermatogenesis. So oogenesis, how can you define this? Oogenesis, the process of oogenesis occurs in the cells of the germinal epithelium of the ovary. Such cells are known as primordial germinal cells. The oogenesis is complete in the following three successive stages. So, primordial germ cells, gametes are derived from PGC, form in the epiplast during second week and then move to the wall of yolk sac, begin to migrate from the yolk sac in the fourth week and arrive in the gonads by end of fifth week. So, the oogenesis is completed in the following three successive stages. Those are multiplication phase, growth phase, and maturation phase. So, this is at stages in oogenesis. So, let's see this picture. So, multiplication phase, the primordial germinal cells divide repeatedly to form the oogonia. Oo means egg. The oogonia multiply by mitotic division and form primary oocyte, which pass through the growth phase. So, it is a multiplication phase. So, growth phase. The growth phase of the oogenesis comparatively longer than the growth phase of the spermatogenesis. The growth phase, the size of primary oocyte increase enormously. Most instant, the primary oocyte of the frog in the beginning has the diameter about 50 mm, but after the growth phase, the diameter of mature egg reaches about 1000 mm to 2000 mm. In the primary oocyte, large amount of fats and proteins becomes accumulate in a form of yolk, and due to its heavy weight or gravity, it is usually concentrated towards the lower portion of the egg, forming the vegetable ball. The portion of cytoplasm containing the egg pronucleus remains often separate from the yolk and occurs toward upper side of egg, forming animal ball. So, let's see this oogenesis processes. So, the portion of cytoplasm containing the egg pronucleus remains often separated from the yolk and occurs toward the upper side of egg, forming the animal ball. In the certain oocyte, amphibia and birds, the mitochondria become accumulate and some place in the oocyte cytoplasm and forming mitochondrial clouds. The nucleus become large due to the increased amount of nucleoplasm, and it's called germinal vesicle. So, the chromosomes change their shape and become giant lump brush chromosomes, which are directly related with increased transcription of mRNA molecules and active protein synthesis in cytoplasm. So, it is a maturation phase processes. So, let's see this, that we can understand. So, here after the meiotic division of the nucleus, cytoplasm of the oocyte divides unequally to form a single large-sized haploid egg and three small haploid polar bodies of polycytes at the end. So, first, maturation division. This unequal division allows one cell out of the four daughter cells to contain most of the cytoplasm and reserve food material, which is sufficient for the developing embryo. First, maturation division. During the first maturation division of first meiosis, the homologous chromosomes of primary oocyte nucleus pass through the firing of synapses, duplication, chiasma formation, and crossing over. Soon after, the nuclear membrane breaks and the bivalent chromosomes move towards an opposite poles due to the contraction of chromosomal fibers. A new nuclear envelope is developed around the daughter chromosomes by endoplasmic reticulum. After the karyokinesis, the unequal cytokinesis occurs and a small haploid polar body polycyte and large haploid secondary oocyte or ootid are formed. So, second, meiotic division. The haploid secondary oocyte and first polycyte pass through the second meiotic division. Due to the second meiotic division, the secondary oocyte forms a mature egg and second polycyte. By the second meiotic division, the first polycyte also divides into two secondary polycytes. These polycytes ooze out from the egg and they degenerate while the haploid egg cells becomes ready for the fertilization. So, a structure of mature egg. So, first, the mature eggs has a cells-like structure and composed of the following. And those are plasma membrane. Plasma membrane, the mature egg is covered by the plasma membrane which is the unit membrane. It is composed of outer and an inner of protein. Both the layers of 58 degrees in thickness. Between the proteinous layers, there occurs is lipodose layer of 68 degree thickness. So, primary egg membrane. In addition to the plasma membrane, the egg of most animals except the sponge and certain coleonterates consist of certain other additional egg membranes. These membranes are known as primary and secondary egg membranes. The primary egg membrane is secreted around the plasma membrane by oocyte itself. In the insect, mollusk, amphibian, and birds, the primary egg membrane is known as vitelline membrane. While in tunicates and fishes, this membrane is known as the chorion. A mammalian eggs contain similar membrane and in them this is known as soma pellucida. The vitelline membrane is composed of mucoproteins and fibrous proteins. The vitelline membrane usually remains closely adhered to the plasma membrane, but its later stages a space is developed between the plasma membrane and the vitelline membrane, and this space is known as perivitelline space. So, secondary egg membrane, it's secreted outside the primary egg membrane by a layer of polycyte cells that surrounds the oocyte by the ovarian tissue before the egg leaves the ovary. It occurs in the form of chitinous shell surrounding the egg and insect and called chorion. In this case, have a microfile for sperm entry. It may secrete female sex hormones as a mammals like membrana granulosa and corona radiata. So, tertiary egg membrane secreted by the oviduct glands of the oviduct absent in placental mammals. Jelly around amphibians' egg. Albumen and leathery or hard shells of reptiles of verb. A tertiary egg membranes are from the oviduct or other ancestry parts of female reproductive system. They may be composed or either jelly coats in the amphibians, albumen and hard horny capsules in elasmobranch species, or albumen shell membrane and calcareous shell in birds. So, the ooplasm. The cytoplasm of the egg cells is known as ooplasm. The ooplasm consists of large amount of reserve food and material in the form of yolk. It also composed of lipoprotein, pigment granules, water, RNA ribosomes, mitochondria, and various other circular inclusion. The peripheral layers of the ooplasm is known as cortex, and it contains many microvilli and cortical granules. The microvilli are formed of the outpushing of the plasma membrane, and they help the transformation of synthesis from the follicle cell to the egg during the development of the egg. The cortical granules are spherical bodies of various diameter. So 88 point in a seed or in eggs and 2 point in the eggs of frog. The cortical granules are surrounded by the unit membrane and originate from the Golgi apparatus. They contain homogeneous and granular acids, mucopolysaccharides, and are present in the eggs of sea urchins, frogfishes, beveled mollusks, some annelids, and certain mammals, but they do not occur in the ova of man, rat, guinea pig, gastropods, mollusks, orodelp, amphibians, insects, and birds. Yolk contents of the ooplasm. The amount of the yolk is in the ooplasm varies from species to species. According to the amount of the yolk, following types of egg cells have been recognized. So 1 microlittle. The eggs with very little amount of yolk are known as microlittle egg. Samples: amphibious, eutherian mammals. So this is alicetals examples. So you need to understand what is the differentiate. So mesolicetal, the ova or eggs containing moderate amount of yolk are called mesolectal ova or eggs. Examples: Petromezon, Diphnoi, and Amphibia. So let's see this picture of mesolicetals. So macrolicetals, the egg with large amount of the yolk are known as the macrolicetals egg. Example: myxine, cartilaginous, and bonifacious reptiles, bird, and monotrimata. So this is the examples in a picture. So let's see this. The egg can also be grouped into two types on the basis of the distribution of the yolk in the ooplasm. Homolicetal, the eggs with eventually distribute yolk contents in the ooplasm are known as the homolicetals egg. Examples: egg of echinoderms. So B, heterolicetal. The eggs in which the yolk is not eventually distributed in the ooplasm are known as heterolicetal eggs. The heterolicetal eggs may be the following types. So, one is telolicetal. Telolicetals, when the amount of the yolk is concentrated in the one half of the bag, form the vegetative wall of the egg. This condition is known as telolicetals. Telolicetal egg may be the moderately telocetals. Examples: amphibian or highly telocetals, hen and other birds. In the microlicetals and highly telocetals eggs, our amount of the yolk is very large and it occupies the largest portion of the egg except the small disc-shaped portion of cytoplasm. The cytoplasm contains the zygote nucleus and it's known as germinal disc. Eggs of fishes, reptiles, and bird. So another one, centrolicetal. In the centrolicetal egg, the yolk accumulates in the centri or the ooplasm or eggs of examples: eggs of insect. Thank you for listening and watching my presentation.